Photosystem 2 functions first in photosynthesis, but it is called photosystem 2 because it was discovered after photosystem 1. It probably also evolved after photosystem 1 as well. In the reaction center complex is the special chlorophyll A that absorbs photons of 680 nanometers, so it is called P680. Photosystem 1 absorbs photons of 700 nanometers, and if you're any good at pattern recognition, you won't be surprised that the reaction center chlorophyll A of photosystem 1 is called P700. So here's a question. Which photosystem accepts higher energy photons? I'm not going to tell you, I'm just going to let you think about it. In the light reactions, there are two pathways that plants use. These are called cyclic and linear. Linear electron flow is the primary pathway used. It uses both photosystems and produces ATP, NADPH, and oxygen gas. So let's take a walkthrough. In linear electron flow, the energy transfers begin with the photons hitting the photopigments in photosystem 2. The excited electrons bounce around until they reach the reaction center and P680. P680 transfers that excited electron to the primary electron acceptor in the heart of the reaction center complex. Now, P680 is oxidized. P680 plus is a very, very potent oxidizing agent. How strong? P680 plus can snatch electrons from water, which releases oxygen gas, O2, as a byproduct. In other words, P680 plus can do what we've considered to be the unthinkable. It can oxidize oxygen. But that oxygen is the least useful part of the water molecules that are split. In fact, as we'll see at the end of this chapter, the oxygen gas is actually counterproductive to the whole process and needs to be eliminated. The useful parts of water are the protons and the electrons, which are full of potential energy. The electrons are put to work in a familiar way. They are run through an electron transport chain similar to the one we saw in the cristae of the mitochondria. The electrons pump protons into the lumen of the thylakoids, which drives ATP synthase, very similar to the ATP synthase that we saw in the mitochondria. In the mitochondria, we call this oxidative phosphorylation. In the chloroplast, we call it photophosphorylation. But these electrons are not quite done yet. There's a whole other photosystem to go. This is photosystem 1, again, because of the order of discovery and evolution. Photosystem 1 is also absorbing photons, albeit of a slightly lower energy or longer wavelength. This re-excites the electrons that came from photosystem 2 and transfers them to the primary electron acceptor of PS1 via P700, which is similar to P680, a special pair of chlorophyll A molecules. From the primary electron acceptor of photosystem 1, the electrons are transferred to a protein called ferredoxin, abbreviated here as FD. From ferredoxin, the electrons are added to NADP+ by the enzyme NADP+ reductase, and the result is NADPH. NADPH is a third electron shuttle or electron bus. The electrons on NADPH have the most potential energy of all the electrons on shuttles that we've seen in the past two chapters. In other words, NADPH is the fanciest electron bus. It has to be, for reasons that we'll see when we discuss the Calvin cycle. This cartoon represents the series of events in linear electron flow. A photon of 680 nanometers energizes an electron, which jumps excitedly up in potential energy in photosystem 2. That electron is put to work through an electron transport chain, this mill making ATP here, where it pumps protons, and that's how it makes ATP. But that electron still has potential energy to be exploited. So another photon of 700 nanometers re-excites the electron, which gets added to ferredoxin and transferred to NADPH, ready to head off to the Calvin cycle. This is linear electron flow, but there is another. This other one is cyclic electron flow. In cyclic electron flow, only photosystem 1 functions. Without photosystem 2, there isn't enough energy to split water, so no oxygen is released. Without 2 photon punches of energy, no NADPH is produced either, but there is plenty of energy to continue to make ATP. Here's a cartoon similar to what we saw with linear electron flow, only with cyclic electron flow. Again, only photosystem 1 is in play here. The electrons will still go to the protein, ferredoxin, but they don't make the cut for the extra fancy bus. No NADPH for these electrons. Instead, they go back to the cytochrome complex where they can still pump protons and make ATP. Cyclic electron flow is thought to be the more ancient process. Some bacteria, namely purple sulfur bacteria and haloarchaea, don't have photosystem 2, so their photosystem is anoxygenic or doesn't produce oxygen. So photosynthesis evolved before there was an oxygen-rich atmosphere, and the evolution of photosystem 2 in cyanobacteria is what produced the oxygen-rich atmosphere that we enjoy today. Plants can also perform cyclic electron flow, and it is thought that it may help protect plant cells from being damaged by the sun. Also, you can never have too much ATP for photosynthesis. As we'll see, reversing the oxidation of carbon dioxide, by which I mean reducing carbon dioxide, is an energy-hungry process. I've been alluding to this throughout this lecture. There are many similarities between the biochemical activities and structures found in chloroplasts and mitochondria. Both use chemiosmosis to produce ATP, even though the energy sources are different. For mitochondria, the energy comes from food. For chloroplasts, sunlight. Both have folded up membranes to increase surface area for maximal efficiency, but there's also a key difference. As you can see in this image, the use of the proton motive force is very similar. Look down here. Electron transport chains release free energy stored in electrons to move protons across a membrane to drive ATP synthase. However, the arrangement is different. In these images, the acidic/lower pH/higher proton concentration is shown in darker gray, and the less acidic/higher pH/lower proton concentration is shown in lighter gray. Note how the mitochondrion produces all that ATP in the matrix, while in chloroplasts, the ATP is produced in the stroma, almost like the chloroplast is functioning inside out. This too is an efficiency measure, because all of that ATP is going to be used in the stroma along with the NADPH with its fancy high potential energy electrons. And that will happen in the Calvin cycle, which is where we are going in part 2.